The title of my talk is Naturalistic Decision Making, and I'm going to start with a little bit of history of science. I'm going to start with the question of that a lot of people walk around in their minds with every day, which is that animals, including monkeys, are just very impulsive creatures. And I would argue that historically, this view can be traced to this one particular study that was done by a graduate student in Richard Herrnstein's lab, Shinho Chung, in 1967. And Herrnstein, of course, was at Harvard and shared a lab with B.F. Skinner, and they worked with pigeons, and they did the first intertemporal choice task, as we understand it today, with pigeons, and published this study. And that in turn led to an explosion of research using that task. And it was really George Ainsley, who was a resident, a psychiatry resident at Harvard, who worked in the same lab, who popularized this work with two papers in 1974 and 75 that really argued that this task gives us a really great measure of self-control and impulsivity in animals, and therefore lets us do a lot of circuit breaking type research in animals on impulsivity. And the reason from a historical perspective this is really interesting is that despite its fame and success as a task, and despite the importance of this perspective and the idea that animals are impulsive, there was criticism immediately that was largely ignored that came from the foraging theorists. Sir John Krebs, of course, and several other people thought that this idea of animals' impulsivity actually is kind of very problematic and needed to be questioned. For example, if an animal is impulsive and engages in costly maladaptive behavior, that should be selected against by evolution. And there's really not many good reasons why an evolved animal should have poor self-control, since it's not obvious why good self-control is more costly than poor self-control. This led to a series of studies, the most important of which in my mind is a study by Dave Stevens here at Minnesota, and his graduate student Dak Anderson, was published in 2001, where they really not just address this idea in theory, but actually use an experimental design to test this idea. And in particular, I don't really want you to pay too much attention to this figure. I just want to show you that they have two very abstract tasks, but one task uses a foraging structure, and the other task uses the more conventional Chung and Herrnstein intertemporal choice structure. And that by just changing the structure of the task, in complete violation of the principle of economics that the method of elicitation should not matter, they got completely different results. And I'm not even going to show the results. I just want to emphasize the idea that you can have animals, when they use a naturalistic foraging-like task, they look perfectly patient. And when they use this laboratory artificial task, they start to look very, very impulsive. And that raises the possibility that maybe the task that Herrnstein and their colleagues and everybody else since then has been using is giving us a biased view of the impulsivity of the animals. My lab has a little bit of a role in this. I put this down here at the bottom. I have a review paper on this whole thing, if you're interested in it. We were able to show that this applies in monkeys. Stevens and Anderson and their follow-up work was on birds, blue jays. We show that this works in monkeys, even with overtraining, even with very sophisticated types of measures, and we're able to link these ideas to the concept of attention and biases in attention, and kind of get some understanding of how they misunderstand the task. And when we use this foraging task with monkeys, that is a computerized task, not totally naturalistic, with two options, but it has the mathematical structure derived from the patch leaving task in foraging theory. Like Stevens and Anderson, we're able to demonstrate nearly perfect patience in these monkeys, the same monkeys that have ostensibly severe impulsivity in intertemporal choice tasks. And I like this story because it illustrates kind of the core motto of our lab, which is that natural is different. When you take the effort to put your animals into a naturalistic paradigm and think about how they interact with the world on their own terms, you're going to get things that are qualitatively different than you will with unnatural tasks or laboratory tasks. This is, of course, a continuum. It's a spectrum, and we've had a lot of success, just like many other people, with laboratory tasks. But we always have one eye on naturalism and one eye on how can we make it more naturalistic, and what is it going to give us that's different. So just to kind of say the same thing in a slightly more verbose way, we can better understand choices, which of course is the interest of our lab, how animals make decisions when we examine them in naturalistic contexts. And just to elaborate even further, we can get new types of behavior, we can get different patterns of old behavior, different types of neural activity, new questions to ask. Before I go and launch into my talk, I'll just give one more example. In the laboratory, most animals are highly overtrained. They've been doing this task for months and months and months. There is no uncertainty about what the structure of the task is. And we don't want that scientifically because that is kind of a source of noise. But in the actual world, in the natural world, uncertainty is like 99.9% of what you have. You just don't know anything relative to what's going on. So there's this constant tension between the need to get more information and the need to get the reward. And the sort of reward information trade-off is just a huge factor in our daily life. It's something that motivates our curiosity, and that in turn has motivated our lab to study curiosity, information seeking, information trade-off. And that's something that unless you specifically design for it or test for it, you just don't get for free in laboratory tasks. So I'm going to tell you about two studies today, or two different directions within the lab that really kind of represent the sort of I would say the first steps our lab is making towards sort of giving up the traditional binary choice paradigm that's been dominant in economics and in our lab, and moving towards the more naturalistic task paradigms. The first one I'm going to talk about is the avatar-based pursuit task, and this is a project that was led by former graduate student Michael Yu, who's now a postdoc at MIT and soon to start a faculty position at IBS in South Korea. And even though it's called the avatar-based pursuit task, we call it the Pac-Man task, and you're going to see why in 1 second. This is the task right here. This is a video of the monkey doing the task. You can actually see the monkey on the left side of the screen right here. He's using a joystick to move a virtual Pac-Man around the screen, chasing a square and trying to capture it. And what's nice, I think, one thing that's nice about this task is because I think it's more natural. You can sort of guess without me having to describe too much what the rules of the task are. There are these triangles occasionally that pop up on 25% of trials that are pursuing the monkey, and he has to avoid them, otherwise he doesn't get any reward. And sometimes he has to choose between multiple different squares that have different values. And I love this task. I can watch this all day. Sort of frustrating. But the monkeys also really enjoy this task too. I'll just say that it's very easy to train them on this task. Once they sort of get the concept of a joystick, they seem to find it very intrinsically rewarding and interesting for them. I'll also just point out that this is not purely natural, right? There's a virtualness to it. There's an abstractness to it. They're still getting juice delivered into their mouth. They're not eating something that they themselves captured. So this is a compromise between the highly abstract laboratory task that's highly diminished and the ultimate goal, which is understanding behavior in situ, in the wild. But we don't do that because this is going to be more tractable. Just in case you need it, here's the kind of our cartoon illustration of the task. There are 5 different prey types that can come up on each trial. You can have 1 or 2 preys. There are 5 different predators, and they differ in terms of the amount of juice that they offer and the difficulty they are to capture. And in these 2 papers that I cite here, there's a bunch of analysis of the behavior. A lot of it's really interesting. I'm not going to talk about it today. I'm just going to illustrate sort of for you one randomly chosen session just to demonstrate that monkeys tend to capture these prey. They think they capture them in like 98% of trials. We didn't want it to be too hard. They usually capture them in 5 or 6 seconds, and they capture them anywhere basically on the screen. They don't really cluster in one corner. They kind of tile the whole screen, although they have these curvy paths, which are very interesting. And I'm not going to talk about the spikes right yet. I will just say that we record brain activity in several brain areas simultaneously. We like to get a lot of data in this task, but right now I'm just going to talk about one brain area, the dorsal anterior cingulate cortex. This is sort of the home base for my lab, sort of the area of most interest to us. It's an area that we call Area 24. It's so poorly understood that that's actually, even the numbering for that area is controversial. And if you're interested in that, I have an annual reviews paper where we argue for that terminology, and we argue for it as a distinct area. It's still probably we're still probably in the minority on that. But it has a lot of interesting links to the emotion system, the cognitive system, and the motor system of the brain. We tend to think of it as something that sort of integrates emotional and cognitive information and generates motor plans, or very abstract motor plans. I'm just going to talk about one possible role of this region today, And I will say that, historically, discussions of the role of this area tend to be very abstract and very high level. They tend to link it to these things that come from like the textbook, the chapter names in psychology textbooks like conflict and executive control, outcome monitoring, and we've argued for a lot of these roles in the past as well. But instead, I want to argue for something kind of much more uh, low level and simple. And what the point that I really want to make is that if you really take the foraging perspective, the naturalistic perspective, our brains are not just a kind of a vat in a jar making abstract, disembodied, disembedded choices with no reference to the real world. Pretty much every decision that we make, at least that monkeys make, has some physicality to it, has some reality to it. So in the classic patch leaving task, which we've studied for about 10 years, in the real world, you actually get up and leave, you get up and move. And so there's a spatial component. And so we believe that space actually winds up being the matrix along which pretty much all of cognition is organized. And that means that the motor system is actually a much more important role than most people think in the world. And so we think that even an area like the dorsal anterior cingulate cortex, which is associated with highly abstract cognitive functions, actually probably has a very deep and dense spatial repertoire, and that's actually going to be the organizing structure of the way that it does these other cognitive functions. So I'm going to, in the interest of not spending too much time on these kinds of things, I just want to tell a brief story and say, here are the responses of 3 example neurons. And what we're going to do is we're going to just look at the spatial repertoire of these 3 neurons. And I just want you to look at the top row on this graph, the row that says self. We're going to use a method that was developed by a graduate student named Kia Hardcastle in collaboration with Surya Ganguly and Lisa Giacomo's lab a few years ago to study the spatial repertoire of non-grid medial-entorhinal cortex cells in the rat. The method that they developed is basically something that allows you to find spatial tuning in spatially selective neurons that does not necessarily look as clean as a place cell or as elegant as a grid cell. It can have any sort of complex blobby shape, but it's a method that allows them to distinguish that between filtered noise. And so we're going to use this method. It's kind of going to be a generalized tool to look for spatial tuning in neurons in parts of the brain that may not have a simple place-like, point-like, or grid-like structure. It may have a more complex structure, but it still encodes plenty of information about the space. What you can see here in this top row is each of these little inset blue-green flags is actually a representation of the pen that the monkey sees. So the monkey is looking at this virtual world where he can chase a circle, or he uses his circle to chase a square and avoid triangles around his screen. The rectangle represents the screen, and the z-axis, the color of it, represents the firing rate of that particular neuron. So if you look at this third, I don't think I have a cursor here, but if you look at this third column here, session 5, cell 16, you can see a big bright spot. So whenever the monkey's Pac-Man symbol is over on the right-hand side of the screen in the middle, you're going to get a lot of activity in this cell, and you're going to get some activity that depends on the position when it's in other areas, but not as much. So what we can see is that these neurons have a selectivity of tuning for the position of the self in this virtual world, and it's this very rich one, and it's actually quite common in these neurons. There's a high degree of selectivity for self. There's also tuning for the selectivity of the prey, and in fact, these are the same neurons in the top row and the second row. So you can see this neuron not only encodes the position of the self, it also encodes the position of the prey, and each of these neurons, when these are very typical neurons, have different tuning functions for the different items. So they're going to respond differently. It's not like when anything enters this particular part of the world, they fire. They differentiate what it is. And then finally, these neurons also encode the position of the predator in space. So they have 3 at least completely distinct maps in the world, and they're overlaid, so they're multiplexed within the same cell. This is not like there's self neurons, prey neurons, and predator neurons. They're multiplexed in a population code that, subject to a linear decoder, can tell the position of the self, prey, and predator with enough neurons. So there's the key point of this slide is there's a rich spatial map in the world in these neurons. And we would not have really even seen it with a simple, a flanker task or a Stroop task or some kind of cognitive conflict task, because we were using an overly simplified task that ignores the spatial reality of decision making. Let's see, just to give a little more details, this is kind of basically an allocentric mapping function. We can also find egocentric mapping functions. So this is the same data. These are just some different cells that I chose because they're elegant. I'll encourage you to look at this first one, session 1, cell 15. By convention, we put up, meaning moving forward. So whichever direction the avatar is moving, if there is a prey, a square, to the right-hand side of the monkey. So if he's moving to the right, then it's down. If he's moving down, then it's to the right, to the left. If it's on his right, and he sees it on his right, if he sees that prey on his right, then it's going to have a high fire rate, especially if it's in the middle distance. If it's really close to him, it's not going to fire too much. But as it moves farther away, you're going to get some activity in the cell. And this has to do with the interaction between the self and the prey. So, and these are independent codes. And again, these are not separate. It's not like there's ego cells and allo cells. These are multiplexed in a single code that is shared across all neurons. So you have this very rich repertoire of information about the physical world in this area that's traditionally associated with these abstract cognitive concepts. And we're not saying that those theories of control or monitoring are wrong. We're saying that they're structured around a highly enriched spatial map that includes both allocentric and egocentric representations. And I'll just talk about one more thing while I talk about this Pac-Man task, which is the question of decision making. So we tend to think of decision making as, where there's two options and you choose between them, two risky options, and which one do you prefer, which one's better, delayed and immediate option. But actually, this is also a decision making task, and you have certain kinds of qualitatively different questions you can ask with this type of decision making that you can't ask with a standard task. So just to give, what to us is the most interesting question: does the monkey actively predict the future? And I was actually involved in a kind of symposium that the Templeton Foundation put on for 3 years, ongoing, about the question of prospection and future looking. And one of the major questions is, to what extent does future anticipation happen in the animal world, in the animal kingdom, outside of humans? We know it's quite robust in humans, but we don't know how robust it is outside of that. And and I tend to believe that animals probably do look at the future, probably do think about the future, but that it might be hard to get with conventional laboratory tasks, and that having a very intuitive, natural task that animals get, seems to be very elementary for them, is going to allow us to uncover this future thinking, this prospection that you might not get with conventional tasks. And in particular, we want to ask the question, if the monkey is using the joystick to control his circle to chase a square, Pac-Man to chase a ghost, in other words, does he point his joystick right at the position of the square? Does he lag that? Does he try to point it at the position of the square but have some delay in his system? Or does he actively predict the future and point his joystick towards the position where the prey is going to be in the future, which would involve some very rudimentary form of future prospection? And I'll just say, the way the task is designed, the maximum speed of the monkey is faster than the maximum speed of the fastest prey. So it would work. In fact, it wouldn't even be too costly to just point the joystick at the position of the prey. But you can get probably about 10 to 15% more reward per unit time if you predict and look forward into the future. And there's a bunch of math in this paper, and I won't go into it, but I will just say that basically the gist of it is, if the monkey is down here where this gray circle is and the square is up there, does he point towards where it's going to be? And the answer is yes. And I'm going to skip all the math because it's not that important for the talk today. And I will say that we tested 3 monkeys in this task, and all 3 of them point the joystick towards the position where they think that the prey is going to be in about 3/4 of a second, something like that. And that actually increases their intake rate, their reward rate, by a pretty decent amount, by a pretty healthy amount, 15% or so over the course of a session. And it seems to be kind of universal. This is sort of something that, when you put a monkey in the chair and you start them on the Pac-Man test, they don't do this. They have like a 0 millisecond future prospection, but over the course of about a month or so of training, this gradually gets longer and longer, and after about a month or a month and a half, it sort of plateaus out at around 3/4 of a second. Not only that, but there seems to be an explicit representation of the future position of the prey in these neurons. Now, I already told you that there's a representation of the current position of the prey. We looked at that. Now we're going to kind of regress that out of the firing and look at residual encoding of the guessed future position of the prey. And we're going to see that there is indeed a map of the future position. And what that means is that if the monkey guesses that the future position of the prey is going to be, if you just look at the top row of images here, session 2, cell 24, if that future position is over to the left on the up part of the screen, then you're going to have high firing. And that's also going to be true for the current cell. So this neuron has a similar map for guessed future position and current position. That is one particular cell, but if you look at the left-hand side of the inset graph in the middle here, you're going to see completely different maps. So the map of the current position and the map for the future position of the prey are entirely distinct, and in fact, somewhat anti-correlated. And what that tells us is that the brain has simultaneous multiplex maps for current and future positions in this part of the brain that, again, we didn't really even think had strong mapping functions or very weak mapping selectivity to it, that actually has a lot of information about the physical world. And it even has information about the cognitive processes that allow for prospection and future thinking to happen. All right, before I conclude this part of the talk and start talking about the monkeys moving around in the big cage, I just want to kind of provide a little bit of context for why this is interesting to us and where we're going. And I want to do that by talking about this opinion paper that I wrote with my former graduate student Michael Yu and with a collaborator of ours, John Pearson. Generally, in the economics laboratory, decision-making is thought of as a choice between two options that are, presented. We never thought of them before. It just came out of the blue, and we decide at one instantaneous point in time. We might have some reaction time, but that there's nothing happening during that time. We make our decision, and then once it's done, we get our reward, and then we never come back to it ever again. And we have argued, and we argue in this paper, that actually those types of decisions, you can find examples of those in nature, in the wild, outside of the laboratory, but they're actually somewhat rare, and that actually the much more standard case, the more normal case, is what we call a continuous decision. And a continuous decision is one in which there's really a continuum of possible options that can be subdivided infinitesimally small, and they take place continuously over time. So you make your decision at time t, and then immediately at time t plus 1, whatever the one unit is, you're going to have another decision to make. And so a classic, the pursuit problem, the prey pursuit problem, of which the Pac-Man task is an example, is just a classic example of this. You just have to decide what exact direction to point the joystick, and then a second later, or a moment later, you have to decide what direction to do it again. Then a moment later, you have to decide what direction to do it again, and it's closed loop. So the options available to you at time t depend entirely on the choices that were made at time t minus 1. And so the decision-making, the kind of algorithms that we tend to think about for decision-making in economics don't actually generalize that well to this much more common case where, you have a, expected utility maximization. It turns out that there's a whole branch of engineering control theory that winds up being much more useful for ways for thinking about decision-making in this more general, more naturalistic case. And so the direction we're going with that in the future, and we have a new R01 to deal with this, is looking at how decision-making, how we can kind of reconceptualize the process of decision-making and optimization using engineering control theory to try to understand decisions of a continuous nature, especially using this prey pursuit paradigm that has been so interesting for us so far. All right, I'm going to take a pause for 1 second and move on to another historical question in neuroscience. And I will ask the question, are monkeys risk-seeking? And I will say, if you don't know the behavioral economic literature, it's pretty well accepted that humans are highly risk-averse. That is, about 80% of people will pay a penalty to avoid a risky option, even if it costs them a certain amount of money. They'll pay probably 10 to 15% premium to do that. This actually seems to be kind of a pretty universal principle in the animal kingdom. There's dozens of animal species that have been tested, and they all tend to be risk averse, at least for reward amounts. When you have gambles for time, things get a little complicated. But with the simple case of reward amounts, animals tend to be risk averse. And in 2005, my postdoc advisor, Michael Platt, and his graduate student, Ellie McCoy, published a paper showing that monkeys are risk seeking. And it wasn't actually even all that important to the study. The study was really about the function of posterior cingulate cortex neurons. But that risk-seeking in monkeys engendered a huge amount of consternation in the field. And Michael's told me at one point that he got more hate mail about that one result than he has about anything else added together in his entire life. And so my first job when I was a postdoc in the lab was to go test and see whether Allie was right and to see how robust this finding of risk-seeking was. And I went and did a bunch of studies. This isn't one of those. This is a follow-up study that we did. But we found that risk-seeking in monkeys is extremely robust in the physiology laboratory. We could knock it around and punch it and do a bunch of things with it, but these monkeys still wanted to gamble. And we've now replicated that in my lab at Rochester, my lab in Minnesota. At least 10 other labs around the world have replicated that basic finding. It's pretty solid result. And yet, despite that, I do not believe that monkeys are intrinsically risk-seeking species. You might think that I would, but I don't. I think that actually, I'm going to make the same exact argument I made at the beginning of the talk, which is that context determines everything. There's something about the physiology laboratory that gives you very weird and special and unusual patterns of behavior. And I've written about what those features are in the past. There's a lot of trials, very small rewards, very fast duty time, and a bunch of other factors. The complete lack of uncertainty in the environment probably seems a factor, the complete overtraining. One way that we tested this is we built a cage in our laboratory at the University of Minnesota and put these feeding stations around it. And you can see 3 of them in this picture taken from inside the cage. And the monkeys in the laboratory were going to do a foraging task. So this is a task using the classic foraging theory paradigm of patch leaving, where if they come up to one of these feeding stations, they'll get a squirt of juice, and then they can get another squirt of juice by pressing the lever again. And every time they do it, they get another squirt of juice, but then that size of that squirt diminishes. And if they move to a new feeding station, they'll get it'll reset to a large value. And we can make some of these outcomes stochastic, which is not usually done in foraging theory and patch leaving paradigms, but can be done very easily, and it doesn't really cause any consternation for the equations of foraging theory. We can take the data from that and elicit monkeys' risk attitudes from that. And what we did when we found we did that is these monkeys who were found to be highly risk-seeking in the laboratory, in the shared task, now in this other context were relatively risk-averse. In fact, they were absolutely risk-averse. And so that tells us that the risk attitude of the animal, the aversion versus seeking, again, like at the beginning of the talk, is highly dependent on the nature of the task, the way we ask the question. And in particular, if we make the task more naturalistic, then we get an answer that makes more sense to us. And more generally, the ultimate goal of the lab, the ultimate goal, I think, of the field, at least conceptually, should be to understand animals in their natural situation. There's this quote by Tinbergen. He says, ethology is interviewing animals in their own native language. That's kind of the way that we tend to think about it as well. We have done some research. This is from a field site in Cayo Santiago in Puerto Rico. We're not really there. We're not really able to collect the kind of data we want there yet, but we're moving in that direction. And so what I'm going to talk about next for the rest of the talk is the Open Monkey Studio project. And this has been a huge, huge undertaking that I started with a postdoc in the lab 6 years ago and has now grown to involve 2 other PIs, Hyun-Soo Park in computer science and Jan Zimmerman in neuroscience. And there are currently 8 trainees working on this project. There's several major grants paying for it. And there have been several other people who've been very important in it in the past. The basic gist of the result of all this work is this right here. This is our Open Monkey Studio hardware. This is the cage that we built. So this is the same cage that I showed in the gambling task, although now we have done 2 things. First of all, we painted absolutely everything that we can green so that we can segment the monkey against the background, just for the exact same reason that Hollywood movies use a green screen. In fact, this is the same exact green that Hollywood uses. It's called chroma key green. And then we've built an exoskeleton around the cage and mounted 62 cameras on that exoskeleton so that we can monitor the position of the monkey no matter which way he turns with minimal occlusion. We can get multiple views with minimal occlusion. Here's just another view of the same thing. We have these barrels. We've since painted those barrels green as well. Every little bit helps. Here's the result of that, of several years of work, including several computer science PhDs. We're finally able to kind of solve this tracking problem. Here's just one monkey moving around the cage. 13 joints tracked. Now we typically do 17. And when I say joint, one is the wrist, one is the elbow, one is the shoulders, the two hips, the knees, the feet, and so on. And then we do electrophysiology. We have a very sophisticated system that was designed by Jan Zimmerman that takes parts from spike gadgets and gray matter. Although Jan has worked with both of those companies to design new products that they don't really offer and that interact with each other and have compatibility with each other and have features that they don't normally have. And that lets us get upwards of 100 neurons at a time safely while the monkey is moving around in this not virtual, in this real environment, interacting with feeders and protected from electrical noise that we need to get data from brain cells. So that actually wound up being relatively straightforward for us, solving that problem. Jan is really a genius at solving these kinds of problems, so he made it seem easy, but we did not spend a lot of time worrying about this, but we did spend a lot of time getting the tracking working. Now, if you're a casual observer, you may think that this problem is easy. You just download Deep LabCut and spend a weekend annotating. The general principle of that idea is true, but the annotation problem is extremely, extremely difficult with monkeys. And there's a bunch of reasons that I list here on this slide. Basically, monkeys, like humans, have need a lot of annotation. You need probably about 10 million images, and generally, as a rule of thumb, it costs about $1 per annotation of labor to collect. So it's going to be about $10 million. And if you know, Google has this tracking system that they have that's kind of the state of the art for humans. I think they paid $12 million for that data set that they need to do that. And so we don't have, never will have $10 or $12 million to spend to get that annotation set. So we got a much smaller annotation set, and then we used a bunch of really clever tricks that Hyunsu Park himself developed, and many of those take advantage of the fact that we have 62 cameras that essentially augment the size of our data set and allow us to get good tracking with our monkeys. And this cartoon here, this is just a monkey. This is a very typical thing. This is a monkey crawling around on the ceiling looking for some grapes that we put up there. This is a very rare pose. You just don't have this with rats and mice, right? They just don't do these kind of weird things. And monkeys do these all the time, especially juveniles. The result of this is Open Monkey Studio. This is our kind of our major product so far. This is a paper that Praneet Bala was the first author on that does 13 joint tracking. As I said, we're up to 17 now. That was not too difficult to get to. And I'll just show this one other cartoon just to illustrate what we can do. This is a monkey. This is actually just the joints moving around with the 62 cameras illustrated. And right now they can move anywhere in this world, and we can get good reconstruction of position. I'll also say, you just have to validate that the system works. You can't just say it. So we have an OptiTrack system, which is the state-of-the-art marker-based system. And there's a bunch of reasons why we don't like marker-based systems, but we did it so we can compare our system to that. And what you can see here is that the blue is the OptiTrack system and the red is our system. And our system gives comparable data, and we think it's actually more accurate because the marker-based system has all this dropout due to occlusion, and we don't have that because we have so many cameras. So we actually think we get higher quality data than the OptiTrack system. So before I show you some of the data that we've collected, I just want to answer the single most common question I have from people, which is, what do you do if you don't have 62 cameras? This is a picture of me 10 years ago at the field site in Cayo Santiago, where very typically you have one camera. You're lucky to have one camera. We are developing a system. This has taken a huge amount of work, and this is a project led by Yuan Yao, who's a graduate student in the lab, that can allow you to track not just rhesus monkeys, but primates more generally with an enormous annotated data set of 100,000 plus images. And we have a model and a variety of backgrounds and a variety of contexts and a variety of poses and, of course, a variety of species. And this is going to be, we're going to be putting this on bioRxiv imminently. We have this database. There's over 100,000 images. We have our model that works pretty well, and we compare it to several existing models in the literature. There's like 3 or 4 of those. And then anybody who wants to can propose their own model, and we're going to have a contest, which is a very conventional way to do things in computer science and not very common in neuroscience or psychology. And we're actually got together some prize money. I think it's going to be $1,000. I'm not sure about that. So if you go to openmonkeychallenge.com, you're going to see the kind of the about the pre-release of that website and of that data set. And I think that's going to be the way forward for getting more general primate annotations. And I'll just say, incidentally, the Open Monkey Challenge and the general problem of solving this was done with a lot of help from the Minnesota Zoo, as well as the primate centers in Oregon, California, and Yerkes, where we've had a huge amount of help just getting images and getting those annotated and getting ethologically valid behaviors from these animals. This is a very typical scene at the Minnesota Zoo, which has the largest collection of snow monkeys in the United States. And so we're able to now, with this system, track these monkeys and annotate 17 joints on these monkeys as they move around the world with the appropriate cameras. That's with a 40 camera setup, but we can now do it with fewer. But the real goal, of course, of all of this is not just behavior. The real goal is electrophysiology. And so this is the first ever data session that we collected with a monkey interacting with our feeders while we're collecting data. I think maybe you can hear this spikes coming out of this system here. There's not a lot here. So this is a monkey doing a foraging task in our laboratory setup while we record brain activity, and these are from the dorsal anterior cingulate cortex. Over the course of a few months, we got over 5,000 neurons recorded in 8 brain areas. And so right now we're in the process of analyzing those. We're also in the process of preparing a second monkey for this. In the field of primate physiology, it's traditional to record from 2 subjects, and there's a lot of good reasons for that. And so we're going to basically replicate what we've already done with our first subject. And this is kind of a heroic work. We sort of just finished the first monkey, and we just have a little bit of data that I want to tantalize you with. So this is a work by graduate student David Mason, who's one of 2 people, along with postdoc Ben Volo, who's really done a huge amount of work getting this up in the physiology part of this project up and running and analyzing the data. And David's project is looking at what I'm going to call place cells, but I put quotes around it because we're not really saying that these are the same as hippocampal place cells. In fact, they differ in some really interesting and striking ways from those place cells. Maybe we don't even necessarily have a good name for them yet. Maybe it's time to come up with one, or maybe we can just call them spatially selective cells. But I'm going to call them place cells is kind of an informal way, just to sort of let you know the way we're thinking about them, is these are cells that seem to provide some information about position in the world. So this is data after we've kind of regressed out all the information about reward that we can and some other variables that I'm not going to go into. This is a view of the cage the monkey's in. Now remember, it's a 3-dimensional environment, but to make it easy to look at, I'm only going to show you 2 dimensions. So we're looking down from above. This is a bird's eye view showing where this one monkey on this one particular day spent his time. He's tended to spend mostly at these 2 feeders. There was only 2 feeders available to him that day. Some days we have 3, some days we have 4, some days we only have 2. And you can see that the neurons tend to fire differentially. We could divide the world into these different regions, and they fire differentially, and they carry information about the position in the world. This is not a typical cell. This is the first cell that we collected. I wanted to kind of show it to you. One thing I will say is that in natural contexts, these neurons tend to fire much, much lower than they do in shared tasks. And we still don't really know whether that's a bias in how we select our cells, because we're doing all our selection offline, or whether that's something about moving around in the natural world. I actually suspect that both are probably true. But I do think that you get much more sparse firing activity when you have natural behavior. And this echoes work that was being done in my graduate lab using natural vision. So that is interesting in and of itself, but the spikes that you do get tend to be very rich and tend to give you a lot of information. This is in dorsal anterior cingulate cortex, and we did this. Of course, we're very interested in comparing this to our data in the Pac-Man task. We also have, here's our first place cell in the orbital frontal cortex. We also recorded in an area called area 13, OFC, on the orbital surface. And this neuron basically only fires when the monkey is on the northeast corner of the laboratory, and hardly fires at all when he's in other positions. And this is, tends to be robust. If he leaves and he comes back, it's not like just doing all this firing at one particular moment. It's spread, of course, around the course of the day. Looking at all the cells in the population, this is very preliminary data. This is a figure from Excel. This is data that was just David just sent me over the weekend for this talk. The positional selectivity is actually pretty strong. You can see statistically significant at p less than 0.05 information using the classic Skaggs definition of place cell selectivity in somewhere slightly over 50% of neurons across all of these 8 regions that we recorded in. The neurons do not do they cover the entire, do they span the entire world? It's not totally clear yet. We're still thinking of ways to do this. We're going to divide the data by day based on the number of feeders that we have. We occasionally had 1 feeder on days when they were trying to do 2 and one of them broke. That's not particularly scientifically relevant, but the 2, 3, and 4 there does seem to be a trend where when we have more feeders, you get greater coverage of the world across the neurons. So if you divide the world into a grid and you ask how many of those grids are represented in the firings of individual cells in each of these regions, you can see there does seem to be greater, richer representation of the world when the task is more complex and there's more feeders, and presumably the cognitive load is higher. There's a bunch of other factors that could be causing that. But this is very preliminary data, stuff we're very, very excited about. We're finally able to start asking these kinds of navigational questions and embedded decision-making questions that we've always wanted to ask. We can finally do that. I'll also say here's a really clean head direction cell that we found in the orbital frontal cortex. So this is a part of the brain that is really associated more than anything else in the neuroeconomic field with encoding of value and comparison of values, and it's often thought to be something that carries very little information about space and the world around it. And we've always believed that as you make tasks richer and more complicated and more naturalistic, you're going to find a much richer map, a much more complicated map of the world within all of the prefrontal regions, much more motoric repertoire in all of these regions. But this is going to be most contentious in the orbital frontal cortex, which is the most associated with pure value. And here's just an example of head direction. So when the monkey is tuning his head towards this one angle, which is 180 degrees, which is by convention, it's basically facing south within the room, he's going to have the highest firing rate. And you're going to see some of that when he's pointing the other direction as well, although not as strong. And when he's at these oblique angles, you're going to see much weaker firing rate. So, looking at the data, it does not seem like there's head direction population of cells and spatial selection of cells. It seems like these are all multiplexed within a single large population of non-categorical functional cells. Finally, I'll talk about Ben Volo's project briefly. This is also very preliminary, but I want to give you a flavor of where we're going with this and the kinds of things you can do. So the behavior of an animal is not just random. They're not just randomly moving their limbs around. That would be convenient for physiologists, but they actually divide their behavior into certain states that are meaningful. And there's a whole set of neuroscience and even statistics that has looked at taking states of animals and automatically detecting them. Josh Shavitz at Princeton, Gordon Berman at Emory, people like that are doing stuff like this. And we want to kind of do that. So this is basically the goal of doing an ethogram, like this person in the slide here is doing, writing down what the monkeys are doing, but have a computer do it based on videos. And there's a lot that goes into doing this, but poor Ben Volo has been working on this really intensely for about a year. It's very strenuous and difficult. and you do a lot of pre-processing. Then you take the data and you put it into a UMAP, and then you can come up with these seemingly meaningful behavioral clusters where you have these different clusters in this abstract reduced dimensionality space that correspond to what we would consider to be relevant, meaningful behaviors. So when this monkey is dangling upside down, that's in one cluster. That's not that common. So it's a small cluster. And then when the monkey is walking, that's very common. That's these clusters that are next to each other over here. And so what we're going to do with this is start asking questions like, what causes, is there a local and global structure? What is the transition function? Is there a Markovian property? What are the kind of basic statistical properties of these states? And then next, what we're going to do, and we have an R01 to look at this as well, is to look at the neural correlates of these states. So how does the brain represent these states? How does the brain deal with this? How does the brain distinguish these from the particular actions that make up the states? And what are the precursors to changes in states? And what causes the monkey to decide it's time to change from walking to climbing? And does the brain anticipate this? What parts of the brain drive this? What parts of the brain follow it? Those are the kinds of questions that Ben V is going to be doing in his work. So that's kind of basically the meat of the talk. I'm going to have 3 slides of future directions. And I will also say that if you're interested in this, if you're interested in working with some of these data, we've collaborating with a few people already. Send me an email and we will see what we can figure out. But I also want to give you some of the directions we want to go in the future. We have a new postdoc who just started this summer, Jeremiah, and his project is going to be doing social decision making. And so these are our first 2 social monkeys that are going to be doing a social foraging task. And they've been doing it a little bit in the cage. Right now, we are just working on really buttressing the segmentation and the identification, the pose identification of pairs of monkeys working together and understand how they interact with each other. And we kind of just barely, as of this weekend, got this segmentation working. So a computer can automatically detect where these monkeys are and can start identifying the pose of those monkeys. In fact, we can do this with our zoo monkeys as well. So we're also starting to take a bunch of high-quality video that we collected at the zoo over the spring and the winter and start to understand how the poses of these monkeys interacting in the zoo relate to each other and what we can say about that. And then I will finally show you this one. I'll turn the sound off. This is kind of the craziest thing that we're doing. We actually mounting the camera on the monkey's head and recording data while the monkey is doing these tasks themselves and integrating the data that the monkey is collecting with the physiology that we're doing in the laboratory. We collected a bunch of data while the monkey was doing this as well and start to try to really take our neuroscience and make it even more first person, even more interactive, even more naturalistic. And that's basically what we're at in the laboratory, and that's the status of things. We just have a bunch of physiology that's just come in and we're starting to analyze it. We're still very, very interested in the question of pose. If you are interested in that, we have a symposium. We're going to just talk about pose in animals. We're going to maybe talk a little bit about electrophysiology, but mostly we're going to talk about the way to automatically detect animal movement. You can go to my website, just haydenlab.com/symposium, and you can get on the email list for that. We won't sell your address or anything. We're just going to tell you what the Zoom link is. And that's October 14th. And that's it.